Hello, everyone. I'm Darren Roblyer. I'm a professor at the Charles River campus in biomedical engineering. And my lab investigates translational, label-free diffuse optics. So what this means is we send light into some part of the body, measure the light that comes back out, and we can say a lot about the tissue based on these measurements. And some of the advantages of this being label-free are that we don't have contrast agents, which means the regulatory burden is low. Devices tend to be cost-effective and non-harmful. And we have many key contrasts. So for example, we can measure the chromophore concentrations of oxime deoxyhemoglobin. We can measure blood flow, and that tells us about tissue function. We can measure scattering and collagen and elastin. That tells us about tissue structure. We also have other molecular contrasts like water and lipid. And we've used these contrasts for a number of clinical applications over the years, some of which I'll show you here. So we have a long-standing study in breast cancer patients where we're monitoring optically breast tumors in neoadjuvant chemotherapy patients, trying to identify which patients are responding to treatment, which patients aren't prior to their surgery. We have a brand new multi-year project in scleroderma where we're looking at this autoimmune disease that causes fibrosis in the skin and internal organs. And we're developing a technique called spatial frequency domain imaging that can measure optical scattering of tissue. And you can see in the scleroderma patient here, the optical scattering is lower than the healthy volunteer. So we're developing this to both diagnose these patients and track them longitudinally as they're getting treatment. We're developing new shortwave infrared technologies, which are wavelengths longer than the visible and the near infrared. These are wavelengths that are sensitive to water and lipid. So we're measuring things like edema and body composition, and we're measuring hemodialysis patients, and we're tracking how water either is removed or not removed from the tissue compartment of these patients, which can affect the efficacy and safety of hemodialysis. We're also measuring the same patient population. We're about to start a study where we're going to measure their cerebral blood flow to try to better understand the cognitive decline that occurs in these patients as they go through months or years of hemodialysis. And then we're taking many of these technologies and converting them into wearable and remote patient monitoring technologies. And I'll focus on two. The first one is related to cuffless blood pressure monitoring, and it's using a technique called spectral contrast optical spectroscopy, or SCOS. Here we use a laser. We send it into tissue. Red blood cells scatter light strongly. We measure the speckle pattern that comes back out of the tissue with a two-dimensional CMOS detector. As those red blood cells travel, they're going to change the speckle pattern. We use this to measure their blood flow. And we can measure this just like your smartwatch measures your PPG or photoplethysmography signal, but it gives you different information. People for decades have been trying to get blood pressure based on the PPG signal, the green light on the bottom of your wearable. But blood flow is completely different orthogonal metric. And we've so far shown that we can get about 30% better accuracy in cuffless blood pressure using this technology. We've just started a new project that's going to take us several years to convert this technology to a wearable. We're working with David Boas in my department, as well as two clinicians, John Foreman and Naomi Hamburg here at the Boston Medical Center. We're also working with Vipul Jatalia to measure hemodialysis patients, and what we found is in patients that have water reductions in their periphery over the duration of dialysis, these patients tend to have fewer adverse events. You can see that in the blue line here. And then, as I mentioned, we're also going to measure these subjects for their cerebral blood flow, and that's going to start shortly. So thanks very much for your time. Here's my email address. I'd be happy to talk to you about any potential surgical applications.